Continuing here with the lens. So the important thing about the lens is that the light will be refracted or bent. So whatever gets projected onto the retina is actually upside down. And the cornea, just let's slide through. It's just a completely transparent kind of outer layer. Then the lens is what refracts the light. And take a look at this. Depending on how thick the lens is, it will refract the light more or less. So here would be a convex lens that is very highly curved right here, and it will bend the light to a certain extent where it then it gets bundled up and creates this focal point. Or here, this lens is much less thick, let's put it this way, and so the focal length is determined by how thick the lens is and how much it refracts the light. Ideally, you want to project or create your focal point, this one right here. You want to focus exactly onto the retina for best results. Now, depending on how far the object is that you're looking at, how far away it is, you need to either flatten out your lens or make it a little bit thicker, and that's done through accommodation. So in accommodation, you will adjust the lens shape so that you can focus on a certain image. So depending on how close the object is, you need to use your near point accommodation. When you get older, these muscles, they kind of get tired. These ligaments that you're using for accommodation of your lens, they kind of get worn out and tired, and then you're gonna lose your near vision accommodation, and so you will have to use glasses eventually. So here the example of how we're using our ciliary muscles to get the accommodation of our lens. Here the lens is pretty flattened right here. So, and here in the next picture, we're going to thicken up the lens, or here it's called rounded, and there you can adjust the thickness of your lens for near vision or far vision, depending what happens. Here, just briefly to mention, if for some reason your eyeball, let's say it is too long, and your lens is trying to focus on an object, but the best it can do is sort of pinpoint the focus here on the spot which is ahead of the retina. Here are your receptors. Now you're focusing right here. That means obviously that your vision is going to be blurry, and that is referred to as myopia and can be corrected with a concave lens. Opposite is true. If you, let's say your eyeball is sort of too short and you are focusing on a spot behind the retina right here, then of course your vision here is going to be blurry again. And so in order to correct that, that's called hyperopia, and we're going to correct that with a convex lens. And your optometrist can tell you all about that. Astigmatism is the condition where the refracting surfaces of the cornea or the lens are not perfectly shaped, and so therefore you get a distorted image. If you've tried that last week with they have these astigmatism charts where you can test for astigmatism. There's a perfectly symmetrical shape, but you don't really see it as perfectly symmetrical because you have a little bit of astigmatism. It's not a big deal. Most people actually have at least some form of astigmatism. Okay, so moving on, how we actually perceive light and how we process that signal, these electromagnetic waves. So here you have the entire electromagnetic spectrum, or a good portion of it. The visible range is a fairly small range right here in the middle. So the visible range goes from about 400 nanometers to about 750 nanometers. So from purple to red, going through blue, green, yellow, orange, and red. Okay, so these are the different wavelengths of light that we can perceive because we have photoreceptors that will then get stimulated with the different wavelengths of light. So let's take a look at phototransduction. So we need to convert the light energy, these electromagnetic waves, electromagnetic energy, into electric signals in form of action potentials. We have photoreceptors to do that. For black and white vision, we have rods. For color vision, we have cones, kind of easy to remember, see from the cones. For color, just kind of make that association. So rods, they're very sensitive, and they will just respond to the smallest amount of light, any light, but they will just give you perception of different shades of gray. Cones, they have those receptors, they can perceive different types of colors. Your best vision is at the fovea centralis. That will be the highest density of receptors. And then at the optic disc, or the blind spot, you have no photoreceptors, therefore no reception. So here you want to ideally project your image, focus it on the fovea centralis, highest density of receptors. That's for your best chance of having the best perception of visual signals. If you are projecting or focusing on here on this spot, the optic nerve right there, the optic disc, and you have no perception, that's your blind spot right there. Okay, so let's take a look at your receptor epithelium. The optic nerve right here, there's a blood vessel right there. So we have 3 layers. The sclera is the outer layer. That's a very tough layer of connective tissue. And then we have a choroid layer. That's sort of this black layer right here. No photoreceptors in here either. No photoreceptors here either. And then you have your receptor epithelium right here. Now, light comes from here. Oops, light will come from here in this direction, so it will have to pass through 2 more layers of cells. So these are bipolar cells and ganglion cells, and it will have to pass through that first before it hits your actual receptor epithelium. These are the receptors right here. This is the receptor epithelium, and then they will synapse back, or toward the front, if you will, to the bipolar cells and the ganglion cells, which then all bundle up and become the optic nerve. So let's take an up closer view here. It's much better right here. So here will be, these are your actual receptors. For color vision, you have the cones, these are in purple, and for black and white vision, you have the rods, these are in yellow right here. These are all rods. All of them will synapse one way or the other to a bipolar cell, and then synapse to a ganglion cell. The color processing, color vision is usually a 1-to-1. So there you have one cone synapsing to one bipolar cell to one ganglion cell, and then that becomes optic nerve bundled up. So there is very good visual acuity. You have this really good perception, but you need quite a bit of light for the cones to perceive anything. Now rods are much more sensitive, but there's convergence. You see that how here these 2 rods, they converge to this bipolar cell, and then this one here on top of that also converges to this ganglion cell. And so now your brain would have no way of telling whether from which exact rod the signal came. So there, with this convergence right here, you will notice that black and white vision is, while it's very sensitive, it's not that accurate. So when things get very dim, then you're losing quite a bit of visual acuity, and you may have noticed that. So here, let's say we are lucky and we are focusing on the fovea centralis, where we have really high visual acuity, high density of visual receptors, light's coming from here. We are stimulating these visual receptors, and then we're synapsing here to bipolar cells and ganglion cells, and they will actually bundle up to become the optic nerve. So again, the processing is, you're going to have your photoreceptors, rods and cones, and they will synapse, they will get stimulated, they receive the signal, translate it into action potentials that then go via bipolar cells and then ganglion cells. And the ganglion cells, these axons, they will bundle up and become collectively the optic nerve. Now, an interesting thing to notice about this is that, remember we said that the image is refracted. So your lens, the light comes through here, it's refracted. And so what you're projecting onto the retina is actually upside down. So basically what you're looking at, let's say if we draw like a stick figure right there. So here's your stick figure. You're looking at this image right here. What's getting projected onto the retina is upside down. You may not believe that because we don't perceive it that way, but actually your brain learns to re-invert the image for you. So while everything is being projected upside down onto the retina, your brain just inverts that back for you to make it right side up. If you looked at the visual illusions that we had that I put into the lab exercise, you may have noticed that your brain makes up a lot of stuff, and that's also true for re-inverting the image for you so that you see everything right side up. But the actual image projection is upside down. And here this slide is addressing the issue of convergence when it comes to rods. So your black and white vision is not you don't have good visual acuity because several rods will synapse to the same bipolar cell, and then several bipolar cells that transport these signals will synapse here to one ganglion cell. So what your brain receives is just information from this ganglion cell. There's no way for your brain to know which exact rod or combination of rods has fired. And so, as I said before, your black and white vision the visual acuity is not there, but it is highly sensitive. And just something that you should really know, the photopigment in the rods is known as rhodopsin. Something I would like you to know. It is a combination of opsin and retinol, where retinol is actually a derivative of the retinoic acid, which is vitamin A. And you may have heard that vitamin A is good for vision. Well, the reason for that is because it's necessary to make rhodopsin, which is the photopigment in the rods. So I wrote this down here for you. The rhodopsin falls apart into opsin and to retinol when the rods are being stimulated, and then you can put the rhodopsin back together from these two components. So here is your write-up about this. Just recap here. The rods function well in low light, but you don't have much of a visual acuity there because of that convergence issue. And the cones have high visual acuity, very true, but you need quite a bit of light input for that. So if it gets very dim, then you're going to lose your color vision. The visual pigments, they convert light energy into changes in membrane potential, and that these actual potentials are then transported to the brain for evaluation. Here again, it's the rods, they contain the photopigment rhodopsin, and that's the one I would like you to know. The cones, they have 3 different pigments that I don't think you need to memorize here. That would be your rods over here, and how they work. There's the rhodopsin right here, and it falls apart into retinol and opsin right here, and these are these little discs that are stacked up in the rods. Now the cones, they have different kinds of color pigments in there. So when the light hits, then in the different photoreceptors, the light energy is then converted into actual potentials. Let's take a look at how that works specifically. So here a little bit more up close, again, same thing. So the rhodopsin is the photopigment of the rods for black and white vision, highly sensitive. As soon as light hits, rhodopsin falls apart very quickly into retinol and opsin at the speed of light, literally. And the cones, we're going to take a look at that next here. So we have these 3 different pigments in the cones for red, green, and blue. So we have blue cones, green cones, and red cones, and they absorb different wavelengths of light better or not so good. And here is sort of how the physiology of all of that works. So essentially in our rod, once the light hits rhodopsin, that leads then to the opening and closing of ion channels, and that ultimately leads to release of neurotransmitter, which then stimulates the bipolar neuron.